So hello everybody, my name is Natalia Rodriguez. I'm postdoc here at University, and today we're gonna do a DNA extraction as we did to get the sequencing of amplicon for BIOservicES project. So for that, the first thing that we need to do in the lab is like wear up the lab coat and also wear up the gloves, and then we're gonna start. Okay, so now, we're gonna use the DNA extraction kit, call it like Power Soil Pro Kit from Kiagen. So beforehand, to follow in the instruction protocol, we need to weigh the soil. But for this project, we use a DNA RNA shield preservation. First step is add 800 microliter of the CD1 solution. I'm going to add 800 microliter. Each sample the 800 microliter of CD1, and now I'm gonna add also this amount to a blank. So for each DNA extraction, we always add a blank, only to be safe or to be sure that that in any flask or bottle we have any contamination, and either like a cross contamination while we are doing the DNA extraction. And now we add to all of them the CD1, and before to go to the beating step, we need to vortex to mix the beads between the soil, so we can see here the soil, the CD1, and the beads that is gonna do a mechanical breakup of the cells. And also with the CD1 we are adding also like a chemical to help this mechanical. Now we're gonna use the bead splitter. We're gonna do at maximum speed during 30 seconds. Okay, so now after the beaten, in the protocol says that we need to do like a centrifugation to separate to separate the CD1 from the soil and also the beads. Now from the super nadan, I'm going to show you how we can see the difference between the CD2. So here we're going to take the super nadan to extract the DNA from there and leave the sediment of the soil. So now we have our sample that we have all the bacteria or the microbes here, and we need to take the super nadan to transfer to another. So for that, the protocol says that we need to take 700 microliter of super nadan and put it in a clean 2 milliliter epi that I just prepared in advance. So I only gonna transfer like 700 with the CD3. The next step is like we need to use a membrane that is gonna catch the DNA. So now the next steps is gonna do like catch the DNA, try to clean the DNA, and the last step is gonna say like to store the DNA. So we're gonna see it. So first we're gonna transfer 650 microliters of the lysate here in the column. We need to centrifuge and repeat this step twice. Okay, so now we finished to centrifuge twice. So you can see we have all the liquid here, and we need to remove this liquid from here. The steam column we need to move to a new, there's like a collection tube that is provided for the heat. So basically we need to move like carefully this column to here. So now, after put it in the new collection tube, in this case it's gray, so we cannot see inside. So now for that, we only need to add the 500 microliters here. That's it. We need to centrifuge again and put it in a new collection tube. Okay, so now after the centrifugation to add in the C5, so we need to remove the liquid. We're gonna do the last step, in which we need to add the C6 solution. So we need to place carefully the column in a new tube. In this one, I recommend you write all the information that we need, with the date of the DNA extraction, everything. So now we're gonna add here so I can show you because then it's gonna be difficult. So we're gonna add 60 microliter of C6, and we need to add over the filter, in the center of the filter, over the filter without touch the filter. This is very important. So we're gonna do for each sample, then centrifuge, and then we have done the extraction. So now we're gonna do the last step. For that, we're gonna add 65 microliter of C6, and filter but without touching. After we add it, so we're gonna go to the centrifugation. After this step, we only need to remove the column from here. Like this, we're gonna have, with all the labeling, we're gonna have our DNA extract here. So once we have this, after this step, we need to measure the quality and also quantify the amount of DNA that we have here. So for that, we should measure in nanodrop for quality, using qubit or picogreen to quantify the DNA concentration that we have, and after that we can do the sequencing.